Thank you. It illustrates the power of serendipity, and I think those of you who are relatively young in your careers, the career message that you might take away from this is to follow your nose, not to ignore stuff that happens or comes up data-wise that is surprising, because it's those surprises that are most likely to lead you to new discoveries. So in the fantasy version of the talk, there's these cells, called CDCs that I'll tell you about, cardiosphere-derived cells. And by predictable and iterative steps, we would get to these new RNA drugs that I'm going to tell you about. But it really happened in a very circuitous way, each one of these inflection points being marked by an unexpected discovery that we followed up on. So CDCs stands for cardiosphere-derived cells. This is a process name. Basically what it is a cardiac stromal / progenitor cell that we first isolated from human tissue about 20 years ago now. And when we first isolated them from human tissue, we had the idea that we would expand these cells ex vivo, that they could be reimplanted into the human heart, where they would tilt the balance towards repair and regeneration and away from fibrosis and heart failure. And they seem to actually do that, but they don't do it at all the way that we expected it to be. They don't work by canonical stem cell mechanisms. At the time that we first isolated them, we initiated the first of several clinical trials. It was called Caduceus. It was published in the Lancet, and that very much followed the canonical paradigm in its design. We infused the cells down an infarct-related coronary artery, and we looked for regrowth of heart tissue. And we were very careful in that study to use autologous cardiosphere-derived cells, because we believed that the cells would lead to progeny throughout the heart, and we didn't want them to be immunologically rejected. But basically, soon thereafter, we turned our attention away from coronary artery disease to something that we first got turned on to because of interactions with one of my colleagues, Ron Victor, who was working on Duchenne muscular dystrophy. But this is an X-linked recessive disorder that affects about 1 in 3,500 male births. It's because it's X-linked. It's very rare indeed in women. And the Duchenne stereotypically presents as a skeletal myopathy. The kids appear to be fairly normal until about age 2 or 3, when they start having trouble standing up from a lying down position without using their arms and struggling a bit. And that's called Gower's sign. And when a parent takes their kids in to see a pediatrician and they describe this behavior, the pediatrician immediately thinks Duchenne. They confirm it, and then both the patient's lives and the parents' lives change forever, because this is uniformly fatal in the 3rd decade now, sometimes in the 2nd decade. It used to be even more aggressive, but we're now better at a pulmonary toilet, and steroids that prolong the lifespan somewhat. But what's less appreciated is that the terminal event, or the terminal pathogenic process for these kids, isn't so much the skeletal myopathy as it is the cardiomyopathy. And it's a cardiomyopathy that is characterized by fibrous and fibrofatty replacement of functional heart muscle. These are late gadolinium enhanced magnetic resonance images in which a normal heart would look like a black donut, but this white part here is hyperenhancement due to the presence of extracellular gadolinium, which makes it light up, and that extracellular gadolinium is reflected with scar. So, and you can see it in this long axis view as well. So what, just very briefly, the pathogenesis involves the deficiency or the absence of dystrophin, which is a kind of a natural shock absorber linking the extracellular matrix to the cytoskeleton. And when that goes off, the cell membrane is more susceptible to little rips that, among other things, let in extracellular calcium. Calcium is on the order of millimolar in the outside space, and normally on the order of micromolar or less in cells. When it gets very high, a bad number of bad things happen. It triggers myocyte loss, oxidative stress, inflammation, mitochondrial inefficiency, and fibrosis. And what we found in MDX mice, which is a naturally occurring model of muscular dystrophy in mice that end up lacking dystrophin because of an exon 23 stop codon in the dystrophin gene in the mouse. Intracardiac CDCs, and this was work that goes back really 8+ years now, increase the number of healthy myocytes. They're anti-inflammatory, they're antifibrotic, they decrease oxidative stress, and they improve cardiac function. That's not too surprising, but the really amazing thing is that injection of CDCs into an MDX mouse heart made the animals walk better and increase the force of isolated skeletal muscle. So something that was injected into the heart must have had systemic benefits. And so there's a bit of a paradigm change that went on in that process of recognizing that. But we've designed this first human trial called the HOPE-1 trial with single-dose intracoronary infusion of allogeneic CDCs in advanced Duchenne muscular dystrophy. The patients already had a cardiomyopathy, and the patients, because they already had a cardiomyopathy, were almost uniformly in wheelchairs. So here's these, basically what we did is we phenotyped. This was a randomized trial. They either got standard of care or infusion of allogeneic CDCs. And we studied late gadolinium enhancement, and you can see from baseline, 6 months to 12 months, at least this patient seems to have less of this white stuff that I attributed to scar earlier, whereas a normal, somebody randomized to usual care over 12 months didn't. This turned out to be statistically significant at month 12 after a single dose of coronary CDCs, which is kind of unusual. Based on what we know now, it was not necessarily something we would have expected. But in a kind of somewhat retrospective analysis, we found a striking benefit also in PUL, which is basically a measure of quantitative measure that physical therapists use called performance of the upper limb that rigorously assesses skeletal muscle function. So this was the first study we did. But then basically what was happening in the background, and Eleni was in the lab when all this was happening. So we discovered that extracellular vesicles, a particular kind of extracellular vesicle called an exosome, was being secreted by CDCs. And this is the way they work. They don't work by the direct implantation thing. Instead, they reside in the tissue for a while, secrete extracellular vesicles. The extracellular vesicles are immunomodulatory, antifibrotic, anti-inflammatory, and a number of other good adjectives that you can attribute to them. And they work that way. The mechanism, we believe, is that they're chock full of non-coding RNA. And the reason that we attribute it to non-coding RNA cargo as opposed to, let's say, proteins or lipids or DNA is that, yes, there are some interesting proteins in EVs, but they seem to be less important because if you very lightly solubilize these EVs with triton, so the proteins are intact, but the membranes are solubilized, and you have RNase in the medium, more than 80% of the bioactivity goes away, even if the proteins are intact. Secondly, they don't have any DNA. And third, they have very little in the way of coding transcripts. It's mostly non-coding RNA. So we thought this was probably the molecular entity that was responsible. And so taking the lessons of recognizing that CDCs secrete extracellular vesicles, we did some studies in MDX mice showing that intravenous CDCs work just fine. And so we designed a follow-up clinical trial. And this was actually a double-blind placebo-controlled clinical trial called HOPE-2 that was published in The Lancet a couple of years ago. And this is now used repeated every 3 months, intravenous, because, why bother to put it in the heart or the skeletal muscle if these extracellular vesicles are going to spread systemically? Just take advantage of that in patients with advanced DMD. And basically what you're seeing here is the natural history or the history over time, over 1 month. First infusion, second infusion, third infusion, fourth infusion, and they infused either placebo, we infused either placebo or CAP1002, which is the proprietary name for allogeneic CDC formulation that was licensed to this company, Capricorn. And so what we found after we broke the blind is a 71% slowing in the decline of performance of the upper limb. Again, these boys were mostly in wheelchairs, so we assessed their skeletal muscle function was by rigorous measurements of what they could do with their arms and hands and so on. Dramatic slowing, which in a composite global statistical test was significant, even though it was only a 20-patient study, 0.0087. But even more striking was the preservation of heart function, which basically stayed completely normal over the follow-up period. And again, some pretty good p-value associated with that. So this is now in phase 3 testing over the composite HOPE-3 trial. Will enroll over 100 subjects with double-blind placebo-controlled, and FDA has told them, the company, that it will be an approvable product for Duchenne if that trial reproduces even half of these effects. And we have no reason to believe that they wouldn't be reproduced. I tell you this because, it'll be something that Robin knows, but not necessarily everybody in the audience. In general, cardiac cell therapy has been a dismal failure. There have been a bunch of clinical trials with a bunch of different kinds of cells, and they've led absolutely nowhere except a few patients have been killed because of highly invasive methods that people have used to try to poke the cells into the heart. And this might actually be the one thing that stands up and shouts at the end of the day. Well, moving on. So that's CDC. So it just motivates you why we got started thinking about regenerative therapy and cell therapy. And then the discovery that extracellular vesicles mediate the benefits made us wonder what is inside the extracellular vesicles that mediates the benefit. I've already told you why non-coding RNAs are interesting. So when we focused on microRNAs, these are the usual suspects because basically we know how they work. And so it's tempting to say, okay, this population of EVs has these 15 microRNAs. Let's see which ones might be responsible for the angiogenic effect or for the antifibrotic effect or so on. So we studied several of these as early as 2014 in the initial description of the role of exosomes. Ahmed Ibrahim was a PhD student at the time and published a few papers, talking about the roles of microRNAs, as did many other people in the field. But we had a wake-up call when we did RNA sequencing of the EV population, the CDC exosome population. This is exosomes from normal human dermal fibroblasts. They're pretty inert. We can put them into the heart or into the body, and they don't do much. But here's the interesting population, and this is only RNAs of less than 210 or so nucleotides. So we're looking purposely at the small RNAs. And what we find is that microRNAs, this blue thing, only 7% of the pie, and any given microRNA is a very small fraction of that. So we're thinking, well, is it plausible quantitatively that these rare microRNAs would be mediating the benefits of the EVs? So it made us think, well, what else is in here? And there were these other slices of the pie that just stuck out like sore thumbs. And one of them was this green thing here, which is a population of RNAs called yRNA. And so we decided to look at obscure but plentiful RNAs. There were a lot of them in these EVs, but we didn't have any clue how they worked to see if any of those mediated cardioprotective effects. And we followed up on that yRNA slice of the pie by looking at all non-coding RNAs in the RNA sequencing to find the ones that were numerically most plentiful in this population of EVs. And one of them really was outstanding in its abundance. We ended up calling it EVYF1, or extracellular vesicle Y fragment 1. And this is the number of counts in one of those RNA sequencing libraries. EVYF1 was present in 370,000 counts. And this is a system in which a typical microRNA was present in hundreds or maybe 1,000 counts. Okay, so this is an extraordinary abundance. And it was the single most abundant non-coding RNA in extracellular vesicles of this type. So we sequenced it, and we found that it was a perfect match. Initial RNA sequencing run had an error, but when we amplified it properly, we found it was a perfect match of the first 56 nucleotides of the human yRNA4 gene. This is the human yRNA4 gene. It's 96 nucleotides. The first 56 nucleotides, perfect match for this EBYF1. And so we wondered, well, what in the world is this? And I'll tell you a little bit in a second about yRNAs, but what we did is we synthesized this. You can actually use solid phase synthesis to make RNAs up to about 80 or 100 nucleotides. And you don't have to use in vitro transcription or bacteria or anything. And we put it in a transfection reagent, a research grade transfection reagent to make lipid nanoparticles. And we put it on macrophages because macrophages tend to be kind of first responders for a lot of the immune responses. And even by then, we knew that macrophages were involved in the mechanism of action of these drugs, of these cells. So we found that EBYF1 lit up the expression of IL-10, which is an anti-inflammatory cytokine. And also, when we did it systemically in vivo in a rat model of myocardial infarction, we found that it decreased, markedly decreased infarct size, but a scrambled version, same nucleotides, but in a randomized order, didn't have any consistent benefit. So it wasn't some kind of, non-specific effect of RNA. So yRNA, what are they? So they're actually fairly plentiful, but also somewhat obscure in their function. The most notorious function of yRNA is that they're complex with Rho and La, which were a ribonuclear protein. They're the first ribonuclear proteins ever discovered in the Jones Stites lab, and they're circulating autoantigens in lupus. And the RNA that complexes with Rho and La is a yRNA. So yRNAs, since then, studies in yeast and elsewhere, there's some thought that they're involved in DNA replication, but their role in mammalian cells is not always that clear. But a canonical full-length yRNA works by binding Rho and La mediate the effects. So what is this thing that we have, the EBYF1? Well, it's a bioactive truncated species. We knew that it's bioactive because we synthesized it and put it in macrophages and in vivo. But the Rho binding site is disrupted. The 3' half is gone, and the La binding site is completely absent. And the structure, when we looked at it and said, is this something we want to convert into a drug? We did a little homework, and we found out that all 20 non-coding RNA drugs that are approved either by FDA or EMA, the European Medicines Agency, and there's a full 20 of them now, are between 18 and 30 nucleotides long, and they're all chemically modified for stability and for decreased immunogenicity. This was a natural compound, not modified, and it was 56 nucleotides. So we started thinking, how can we make it more friendly to the FDA when we submit it in a regulatory document? So we wanted to make a viable non-coding RNA drug using EBYF1 as bioinspiration. And we also wanted to know the mechanism of action. Here, again, the drug development paradigm is completely different than that which is used in all of those 20 non-coding RNA drugs. All of those are siRNAs or ASOs or aptamers that are designed usually to interfere with some molecular pathway. The one I know the best, Eleni will know about the one against amyloidosis. The one I know the best is called inclisiran. It's a non-coding RNA that targets PCSK9, and in so doing, it has this massive anti-cholesterol effect. So instead of taking a statin every day, you can get a shot of inclisiran every 6 months and have a dramatic decrease in your circulating LDL. That's an approved drug. That paradigm is becoming more and more popular, so that now recent New England Journal paper on angiotensinogen, siRNA, subcutaneous, the same company, Alnylam, and you can get a pretty good antihypertensive effect that lasts up to a year after a single subcutaneous injection. So that is all fine and well, but in designing those drugs, you're limited by your knowledge of existing molecular pathways. Here, we took the opposite approach. We mined these extracellular vesicles for things that were plentiful, asked, are they bioactive? If they were bioactive, we tried to figure out how they worked. And it's not a trivial pursuit, as you can imagine, but we've gotten there with a couple of them. So first, for for EVYF1, to make a drug out of it, and we ended up making a drug called TY1, therapeutic YRNA1. We started, here's the EVYF1 sequence, and we got a clue as to where to focus in because there was another very, fairly abundant little fragment called NT4 that was basically a 24 nucleotide subset of EVYF1. So we knew that we could hone in on this as our region of interest. We started mutating it so that it's no longer a product of nature. And we started putting in backbone modifications and methylations chemically. This was all like the work of 2 people in the lab. If a drug company were doing this, they would have made 3,000 drugs and, had 100 people working on it. But this is just, backroom tinkering. But in so doing, we ended up with something really quite interesting. This one, TY1, ended up being, had a mutant in the first position and has 6 backbone modifications, the so-called locked nucleic acids. And TY1 in the screen and macrophages was at least as good, if not better, than the parent compound in lighting up the expression of the anti-inflammatory cytokine IL-1, IL-10, and it also is much more stable and much more resistant to RNase digestion. So I said, okay, let's run with TY1 and see if it's good. So we put TY1 on bone marrow-derived macrophages from rats, did global transcriptomics, and it looks kind of like the flag of Ukraine or Sweden, but basically each one of these lines is a transcript. And it's not just a random bunch of things that get upregulated or downregulated. There's a striking preponderance of immunomodulatory pathways, and the one that lit up the most was the DNA damage response. And I'll tell you all about the DNA damage response in a minute. But basically, we chased this by trying to get to a specific, we did both RNA protein interactions, and we did RNA-RNA interactions by 2 different methods, a bait capture and a probe-based capture method, and pulled out TREX1. And we did in work that I'm not showing you here, but we just posted this on bioRxiv, and it's in revision for publication if you want to read the whole story. But we did a whole lot of validation to make sure that none of these other things was being pulled out as uniquely as TREX1. So what is TREX1? Well, TREX1 is 3' DNA exonuclease. And it is an interesting molecule because it sits right at the gate as a gatekeeper for the cytosolic DNA damage response. As nuclear DNA is stressed in common conditions or in the extreme and radiation injury or whatever, you get leakage of cytosolic DNA fragments into the cytosol, and TREX1 tries its best to degrade those. TREX1 probably originated not to cope with nuclear DNA or mitochondrial DNA in the cell, but rather with DNA viruses to get rid of them. But if TREX1 fails to digest the cytosolic DNA or somehow bypasses TREX1, you get activation of this very strong, very primordial, goes back to the Precambrian era in evolution, pathway called gas and sting. And this cyclic GAS makes a cyclic dinucleotide, which then turns on this interferon gating gene sting, and you get basically a lot of interferon secretion and senescence activated program, and things get really ugly really quick. So TREX1 is known to be involved in disease, and that TREX1 deficiencies lead to lupus and other autoimmune diseases. But nobody's ever explored the possibility that TREX1 upregulation might actually be a valid therapeutic principle. So we followed the conjecture that maybe a compound that increases TREX1 activity will have broad disease-modifying bioactivity. And so we asked, is this interaction, this RNA-RNA interaction, actually leading to an increase in TREX1 expression? So the first thing we did is we did luciferase constructs, which ligated either the 5' untranslated region of TREX1 to luciferase, or and this is a control in which we scrambled the UTR, or we put in the 3' untranslated region luciferase. And we did these because precedents in the literature made us think that if there's going to be an RNA-RNA interaction that actually increased or decreased expression, that it would be at the UTR level. And sure enough, the luciferase signal lit up with TY1, but not with any of these controls and not with the 3'. And when we put TY1 on a macrophage, it increases the expression of TREX1. And so, okay, that's in bone marrow macrophages. What does it do in vivo? So in a rat myocardial infarction, we ligate a coronary artery for 45 minutes and reperfuse. If we infuse TY1 20 minutes after reperfusion, okay, you don't have to know that the person's going to have or the rat's going to have a heart attack. You do it afterwards. We see striking preservation of infarct size goes down, circulating troponin I levels go down. This is phospho-2 HAX. This is a DNA damage marker that's commonly used in immunostaining, and you can see that's way down. It's a marker of the DNA damage response. And even though I'll tell you later that we think the action is actually in the Tregs and the macrophages, not in the tissue, we're still able to resolve an increase in, not in the tissue bulk, we're still able to resolve an increase in TY1 both at the message and at the protein level. So in a longer term model of myocardial infarction, again, 20 minutes after 45 minutes of ischemia, but now we wait 25 days for the terminal phenotyping. Ejection fraction goes up, fractional shortening goes up, and interior dimensions, both in systole and diastole, go down, so there's less adverse structural remodeling. And you can see in Mason's trichome stains that a scrambled version of TY1, same nucleotide content, but in a randomized order that has no homology to the mouse or the human genome, that doesn't do anything. Again, a very important RNA control, but TY1, saves the day. And there's a little hint that maybe the scramble is somewhat bioactive, and in some of our assays, this particular scramble does give us a little signal, and we've randomized it further in an effort to get rid of any remnant bioactivity. So, but we're confident that it's not a non-specific effect. So why do we think macrophages are involved? I've told you in a way, by way of preview, that we think the action is in macrophages. Well, the first thing we did is we depleted macrophages and then gave TY1 in the same paradigm that I've shown you earlier. And one way to deplete both circulating and tissue macrophages is by using these liposomes called claudronate, which basically are eaten by the macrophages and it kills them. And so we pretreated the animals with claudronate. This led to more than 50% tissue macrophage depletion. And sure enough, when we do this, TR1 is no longer cardioprotective. Here's the infarct size with just vehicle, and here's the infarct size with claudronate, no protective effect. The other thing that we thought was interesting is, and this is super interesting, is that we can get the benefit of TR1 just by infusing macrophages at the same time that we normally would give TR1. Instead of giving TR1 intravenously, we give them intravenous macrophages that have been exposed to TR1. This is macrophages that are involved to, these are two controls, macrophages exposed to vehicle, macrophages exposed to scrambled version. These are macrophages in which we purposely upregulated TREX1, the putative target. And this is macrophages in which we put in TI1 and an siRNA against TREX1. So if our fantasy is correct, that macrophage TREX1 bioactivity is where it's at, TI1 should work, scramble shouldn't, vehicle shouldn't, TREX1 should work, and this should attenuate the benefit of, and we infused, I think, it's in the paper, but I think we infused 50 million 50 million macrophages by adoptive transfer intravenously. And yeah, sure enough, vehicle, big infarct, scramble, big infarct, TI1, massive cardioprotection, Tregs, conditioned macrophages, very good effect. But if we add an antisense or an siRNA against the TI1, against TREX1, TI1 is no longer bioactive. So macrophages do it. So this we thought was a very powerful demonstration that that the macrophages are where it's at in terms of the mechanism of action. So a mechanistic summary right now is that we put in TI1, it gets uptaken by macrophages where they upregulate TREX1, and TREX1 limits DNA damage and its nefarious effects and leads to infarct size reduction. So if that's all true, there's no reason to believe that TREX1 is somehow, or TR1 is somehow going to be specific for ischemic injury. Ischemic injury is just a convenient form of sterile inflammation. It's been associated somewhat with DNA damage, and there's every reason to believe it could work in that situation. But what about other diseases? Heart failure, preserved ejection fraction, which is a metabolic syndrome in which the heart becomes stiff and diastolic function is impaired, or Duchenne muscular dystrophy, which I've told you about a bit already, or scleroderma and systemic sclerosis. So we've done testing in all of these in vivo models, and I'll show you the data. So to test HFpEF, we used 2 different rodent models. This is one of them. We also got beneficial effects in the other model, but I'm not showing you those data. This is a so-called 2-hit mouse model in which the animals are made hypertensive by feeding them a nitric oxide inhibitor, L-name, and they're given, an unhealthy diet. Within 5 weeks, they become obese and somewhat hypertensive and develop diastolic dysfunction. And at that point, once the syndrome is already manifested, we randomize the animals to receive either vehicle twice a week or TI1 twice a week intravenously, or once a week, or once every 2 weeks. And then we followed diastolic function, treadmill distance, circulating BMP levels, and circulating IL-6 levels. And, you can just eyeball this and see that something very special is happening with TI1. This is the natural history of the disease. Diastolic dysfunction, E over E prime, gets bigger. Any dose of TI1 fixes that. Distance run on a treadmill goes down with the full-blown syndrome as manifested with vehicle. Once every 2 weeks, TI1 doesn't do much, but once weekly or twice weekly, big effect, totally normal. Circulating levels of BNP, even a sprinkle of TI1 seems to bring down the BNP levels, and the circulating inflammatory cytokine IL-6 goes way down with any dose of TI1. So this is an interesting effect because it's not just kind of delaying the onset or blunting HFpEF, it's curing it. And these animals are still just as fat as they were. It's not a semaglutide effect. It's another effect. It's on top of no weight loss. So what's going on in the tissue? And this is also extremely interesting. So in HFpEF, a lot of inflammatory and senescence-associated genes and interferons and all sorts of things go up, and they go up in a coordinated way. But if you give them TI1, this is bulk RNA sequencing in the myocardium, TI1 fixes it. Every single gene comes down in a very coordinated way. It's really very striking. It's not like, oh, we're fixing one gene. No, we're fixing the whole thing. It's amazing. Okay, well, I've told you that MDX mice are a naturally occurring model of Duchenne muscular dystrophy, that they develop a cardiomyopathy and a skeletal myopathy. So we decided, let's take 10-month-old MDX mice that already have a full-blown cardiomyopathy and a skeletal myopathy and give them twice-a-week therapy, either with vehicle or TI1. And rest assured, we've also thrown in a scramble and it doesn't do anything. But here's just the vehicle data. So after this 8-week period of twice-weekly therapy, we found that EF goes down a little bit over that time, natural history in the vehicle group, but goes up or stays the same in TI1. the treadmill distance that the animals can go. We think this is more reflective of skeletal muscle function than heart function, but it may be both, goes up. Isolated torque of the isolated skeletal muscle. So there's no ambiguity whether this is a volitional effect or whatever. Almost doubles skeletal muscle. Fibrosis in the heart, here's vehicle, TOI1, much less fibrosis. Here's the tibialis anterior muscle in the leg, skeletal muscle, down, and you can see there's an antifibrotic effect in both of those. So this is not a full phenotypic reversal. We don't expect necessarily a full phenotypic reversal in a monogenic disease. You know, it's not like we're correcting the dystrophin. We're just correcting some of the downstream worsening of the, but this is a striking benefit. Okay, finally, scleroderma and systemic sclerosis. So, because some of you may not have heard about this, it's an autoimmune disease that affects mostly women in their mid-years of life. And there's 60,000 new cases per year in the United States. It's rare, but it's not that rare. Almost every major academic medical center has a scleroderma clinic. What happens is the scar tissue deposits thicken and tighten the skin. And that leads to inability of patients in the extreme to even open their mouths. And it leads to almost impossible use of the fingers because the skin gets so tight. That's bad enough, but about a third of the patients develop systemic fibrosis, and then it's called systemic sclerosis. And that affects the heart, the lungs, the kidneys, and the GI tract. And when the heart is involved and the lungs are involved, this disease has about a 28% 10-year mortality. So it's not a benign thing. It's absolutely incurable. There's not a single specific drug or therapy that's been found to work in this disease. Caveat, there's one of the monoclonals that's used in pulmonary injury has some effect on things like VLCO, some things you can measure about pulmonary function, but no effect on mortality in patients who have lung disease. So with the sole exception there, nothing's worked. So we thought, okay, this would be a really interesting disease to see if this works. So we implemented 2 models of scleroderma. One of them is shown here. It's a subcutaneous bleomycin model. And this model is induced by giving subcutaneous bleomycin for 4 times a week for 3 weeks. And by this time, the syndrome is fully manifested. Then we stop giving bleomycin and we treat twice a week with either vehicle. Controls means they didn't get any, they're just normal animals that got an injection of saline instead of bleomycin. But the ones that got bleomycin, we gave them either vehicle twice a week or scramble twice a week or TR1 twice a week. And you can see when the vehicle ones, that's the natural history. So at 7 weeks, the skin thickness is way up, and you can measure skin thickness from the epidermis through this fatty layer down here. That's called the dermis. It's blue, thin in control, thick in vehicle. Scramble doesn't do anything. TR1 pretty much normalizes it. And cardiac fibrosis, likewise, is almost gone after TI1. Here, the scramble seems to have a tiny bit of bioactivity, but you can see TI1 really kind of fixes it. And yeah, so I mean, it's an incredible effect. So is the efficacy in systemic sclerosis related to this mechanism that I told you about involving TI1 and TREX1? So here's, just to recap, cell stress occurs. There's DNA damage with cytosolic DNA floating around. Meanwhile, TY1 has come in and upregulated TREX1, which mitigates the DNA damage and prevents or attenuates the development, the activation of gas, which produces this 2', 3' cyclic GAMP. That molecule signals the activation of STING, which makes interferons and inflammation. Okay. So here's the first consistency check. Well, if we deplete macrophages the same way that I told you earlier, using claudinate liposomes, there's no longer any antifibrotic efficacy of TI1 in mice in the skin. Okay. Second, when we look at markers of DNA damage in the skin, in the mouse, TR1 increases Tregs, here's that. And it decreases, dramatically decreases, IRF7, which is a downstream interferon, and also IL-6, which is another kind of cytotoxic cytokine. And not only makes it better, but it makes it even somewhat better than control, which is really quite striking. And now human data. These are now human SSC patient macrophages that are exposed to TR1 in vitro, compared now to a scramble. This is the elaboration in the cells of 2', 3' CGAMP, this thing. It's cut down by about half, very significant effect. And you look at this downstream interferon beta, also decreased. And finally, if you look at the conditioned media that's secreted by human SSC macrophages, they secrete much less of this interferon cytokine response, CXCL10. And so another consistency check on that. And you can tell, by the way, this is cut and pasted, that this was from an R1, right? This is all the preliminary data set. So the next steps to the clinic, pre-IND, we submitted it to FDA. In an interview with the founder, or the first successful CEO of Alnylam, he said casually, it takes $1 to $2 billion to develop a non-coding RNA drug. So here we've gotten to pre-IND, to the first regulatory filing on the, little old lab. It's not like we hired one extra technician to do toxicology studies. We need to do clinical trial grade scalable manufacturing, but the good news is we've gotten a vendor to agree to do this for us. And then we do first in human studies. So the FDA, we're waiting to hear from the FDA. They could laugh us out of the room. Who are we? What's this compound? How's it work? Why is it safe? But we'll see what they have to say. So let me just review the discovery arc. We started with hearts. We made CDCs, goes back 20 years. These CDCs have a life of their own with clinical trials. We made EVs, and the CDC EVs are now also on a clinical trial pathway for ventricular tachycardia associated with ischemic cardiomyopathy, where we anticipate first in humans studies maybe beginning 2 years from now. From that, we isolated EBYF1, and that was bioinspiration for TY1. So just to recap, we started with cell therapy targeted at the heart, first autologous and then allogeneic. We identified extracellular vesicles as the mediators, and that led us to use intravenous cell delivery in humans. And then we started mining the EV contents to identify defined factors and use those defined factors as bioinspiration for new chemical entities, leading to next generation therapeutics. So is this a generalizable drug discovery paradigm? It is. We have 4 entirely different families that we've mined and created this way. Here's the one you've heard about. The natural thing we're calling, we came up with a more logical nomenclature. YREX1 is now what we used to call EBYF1, but there's something further down, not overlapping at all, on the YRNA4 gene called YREX2. YREX2 is also, we've also made synthetic derivatives of it, and it works dramatically well in MI, but by a completely different principle. And that paper just got accepted in the European Heart Journal, so look for that. YREX3, another cardioprotective. tRNA, TREX1, not TREX1, but TREX1 is being developed now for MI and for DMD, very striking bioactivity in Duchenne. And then a long non-coding RNA, BCYRN1, we've made 3 synthetic derivatives, one of which is these are super interesting because they selectively increase regulatory Tregs in vivo and in vitro. So yeah, I didn't mean to get out of it, but my acknowledgement slide, I don't know if I can you help me just put up the acknowledgement slide? I'm sorry, I inadvertently clicked out. For that. Excellent, thank you. Yeah, so I'm glad, thank you for that, because this is the most important slide. The folks who collaborate directly on the sort of the second half with the RNA development are Ahmed Ibrahim, who, as he told you earlier, when he was a PhD student of mine, recognized the role of extracellular vesicles. He went to work in biotech industry for a couple of years, thought it was not for him, came back, and he's been very successful as an independent investigator. Kazu did the HFpEF experiments. Ali did a lot of the TOI1 mechanistic experiments. Russell and Alice did the Duchenne studies. Mike Jones did the scleroderma in vivo studies and the superb in vitro studies, and co-developed the long non-coding RNA that I told you, the derivatives increase Tregs. And we've gotten some funding and we've taken advantage of our friends. Thank you.